filling up.
Hi, Frans. Okay, good morning, good afternoon, and good evening to all our attendees today as you're coming in. So I'll just give a few seconds now to allow you to join us. You're very welcome to this evening's very special event as part of Rare Disease Week, where we are celebrating the Retina International Lifetime Achievement Awards, which will be given to our awardees this evening, Professor Frans Cremers and Dr. Frank Brunsmann. To open today's proceedings, I'd like to pass over to CEO of Retina International, which is Avril Daly. Avril, over to you.
Thank you, Fiona, and thank you to everybody for taking the time to share in a celebratory event tonight. We don't have so many these days, so it's really great for all of us to get together to celebrate two wonderful achievements in the area of retinal research. Also Rare Disease Week, as you know, and what better way to celebrate a week like this than to really highlight the real fantastic example of patient partnership with research. I've worked in the retina space myself for 21 years and in other disease areas as well. And I think this example is one of the best I have ever come across and one of the most inspiring. So it really gives me personally, as a person living with an IORD and a person who's worked in this space with many great people over the years, the opportunity to be with you here this evening and listen to the story of these two great gentlemen and their experience and their wonderful legacy that they have worked so hard for us, the patients. And that's really truly inspiring and an exemplar for Rare Disease Week. So I'm going to pass over to Fiona, who's going to be our Master of Ceremonies for the evening. Thank you, Fiona.
Thank you very much, Avril. And thank you for opening today's event for us. I'd like to now introduce Frans Bedora, who is the chair of Retina International. Frans will be introducing today's event and today's awards. Frans.
Yeah, good afternoon, good evening, good morning, wherever you are. To all of you, it is really my great pleasure to let me welcome all of you to this wonderful event, most unique due to the pandemic situation. Normally we will have that ceremonial event on our Retina International World Congress, which takes place every 2 years. But due to this terrible COVID-19 situation, we have had to postpone the last year's meeting in Iceland, and we decided to award, nevertheless, these very extraordinary, amazing people in our retina field, Professor Frans Cremers and Dr. Frank Brunsmann. And I really have to say that I knew both of them very well. Frank Brunsmann, he was one of the first persons I met in my personal retina career, so to say. I think it was at the end of the 80s, Frank, when I remember correctly.
Right.
And Frans, I think it was, I thought about it over the last days. I think it was 1994 in Jerusalem at the RD meeting. Maybe you was there, or was it in Dublin, 1990? I couldn't remember exactly, but it's a long time ago.
Yeah.
And both of them really amazing persons, and it is a great pleasure for us, for Retina International, for the whole Retina International family, to award them with this Lifetime Achievement Award. And yes, I'm really excited, and I want to make it brief, that we have room for the story of Frans and Frank. They will tell us after the ceremony, and I will hand over back to Fiona. Thank you very much.
Thank you very much, Frans Bedora, the chair of Retina International. And indeed, Frans and Frank will be sharing their story, and truly an incredible story at that, after the presentation of these awards. So Due to COVID-19, the presentation of these awards is now online, but we are delighted to use this opportunity to bring the Retina international community together. And to present today's awards, we're delighted to welcome back former president of Retina International, Ms. Christina Fasser. And Ms. Christina Fasser will be presenting the awards now to Professor Frans Cremers and to Dr. Frank Brunsmann. Christina, over to you.
Thank you very much, Fiona. It is my great honor and privilege to present today two friends, long-time standing friends and people that accompanied me during also these many, many years of Retina International. The first I wish to present to Professor Frans Cremers. Professor Frans Cremers started to work in inherited retinal degenerations with his PhD thesis and is stuck until today with IRDs. Professor Cremers has brought a lot of knowledge about IRD genes to the community and thus helping to discover the genes, to characterize the genes, and paving the way to treatments. Professor Kramers is an extremely generous personality. He is also very humble. He would never say, I'm professor, but he was, in my opinion, he is professor. And he was always generous in advice, in helping. And he always said to me, what I'm doing is to help to treat blinding diseases. And he also did it not only in working in the lab, publications, he also did something very important. He taught many, many young students, and he gave these young people the idea that working in IRD is a fantastic opportunity and would bring sense to scientific work. I'm very pleased to present you, Frans, with this Lifetime Achievement Award. I know you still have a lot of in your head and things to come, but take it as a recognition of the patience and the thank and gratitude that we have for all what you did, but above all for your humanity, for your friendship, for your generosity and patience to ask any question and make it so stupid as ever. You always give the feeling you are for us here and you work for us. And that's something of the most important things I ever saw. And thank you so much, Frans, for all you do for us. And I hope we will have the opportunity at a personal meeting in the not so distant future to celebrate this once again in person. But now, please accept the Lifetime Achievement Award of Retina International, of the members of Retina International and all the patients represented.
Thank you very much, Christina, for these wonderful words. So at the end, I will tell a little bit more. Thank you to all. But for now, thank you very much for these nice words.
You're most welcome, Frans. And it's a funny thing. We have today Frans, and Frank, which are very similar names and have same basis. And it is my privilege now to present the Lifetime of Achievement Award to a very special person, to Dr. Frank Brunsmann. Dr. Frans Brunsmann was diagnosed with choroideremia when he was 19 years old. And as many of us, we know that this news is dreadful news. It's shattering news, and suddenly all the things that we take for granted are not granted anymore. This happened also to Frank. And I remember at a certain point he told me, my doctor told me, you will be blind at 30. which luckily enough was not true. But you can imagine what this means for a young person. Frank, he had his fight with his words of his doctors, but he took out the best. He started to think, what can we do? What can we do to advance treatment? How can I convince people to do something? And for all of you that are younger, at that time, there was no internet. A letter to the United States took 1 week. Phone call costed for 15 minutes more than $10. And the dollar was 4 Swiss francs at the time. Today it's 88 cents. And he did it. He asked the researchers, what can I do? And he didn't wait that everybody else does it. He did it himself. And he is my idol for how patients can take a bad news into good action. He worked very hard. He changed his career. He went into research himself, always in mind, how can we best find enough patients to find the deletions for choroideremia and passing cells the way to the treatment. And he was not, when he had that news in the 90s, he didn't stop. He continued to advocate as a patient, to bring forward what we need to have, and also to pass the way that the future of today's young people receiving such a devastating diagnosis have a brighter future. Frank, I would like to present you with this Lifetime Award in recognition what you did as a patient advocate, always apart from your work and aside of your family life. You are a role model for many. Sometimes we were discussing hard. You were stubborn, but stubborn in a very good sense. And I'm really proud that I can present you today with this Lifetime Award, with the gratitude and thankfulness of the Retina international community. And you know, there are many friends watching this webinar today and hoping that we will have also the possibility to celebrate this in person at the next Retina International Conference. Frank, I'm very glad that you get this award.
Thank you very much, Christina, for these wonderful full words that went directly into my heart now. Yes, thank you.
You're most welcome. Good luck, Frank. Back to you, Fiona.
Thank you, Christina. Your presence and your warm words always add such depth and intimate knowledge to our community, and we are truly thankful for you joining us today despite you feeling under the weather. So really, thank you so much for your presence and your contribution.
It's a pleasure. Thank you.
I'd like to move forward now into a really exciting part of our event today. And is Frans Cremers with us?
Frans lost the conference. I think he will be back very soon.
No problem at all. So today,
We lost him.
we will be sharing with you a story, a Q&A interview with Frank and Frans once he is able to rejoin us. So this is a story of a real powerful interaction between a patient, Frank Brunsmann, and researcher, Frans Cremers, and the true powerful impact these relationships can have on research. I'm hoping that Frans will join us very shortly. As we wait for Frans to rejoin us, perhaps I'd like to invite anyone from the audience who would like to share any words of special thank you to maybe to Frank, as Frans is not here at the moment. So please feel welcome to raise your hand with, is it Alt-Y, is it to raise your hand, and I will invite you to unmute yourself. So I would like to invite Eric Hartman to unmute yourself. Welcome, Eric.
Hello.
Hello, Eric.
Oh, good. Well, good evening, good morning. I just want to extend from the Choroideremia Research Foundation. Excuse me. Sorry. This is an amazing moment for the global choroideremia research community. Dr. Kremer and of course, Frank, your example of what it means to advocate as a rare retinal disease patient and what you have been able to do has been a true example to so many of us. And from our global community, I just want to wish our congratulations to each of you on a very, very well-deserved award. Thank you very much.
Thank you, Eric. Thank you for sharing your words. Frank, did you want to respond briefly?
Yes, thank you very much. And I hope that the words of role model is not only said like that, but there are, and I'm very sure that there will be a lot of good other future role models here around, huh? Yeah.
Absolutely. Onwards and upwards. I'd like to invite Claudette Medifint now to please unmute. And Claudette would like to express, I'm sure, some words of thank you.
I would like to speak first to Frank because I met him first. When I met him, he was a lot younger than he is now, and he had a lot more hair and probably also a lot more vision. But I see what has not dimmed is his sense of humor and his tremendous energy. He was always almost like one of those Tesla machines, you know, with the electricity sparking out of him. And he was such an inspiration to all of us as patient advocates who really did not know what we could do. And I think it's only, he knew then instinctively what has taken us years to realize, that the power is in the patient's hands. And he showed us what actually could be done. And for that, I thank him. And I don't know if Frans Cremers is back yet. I never really knew him that intimately, but he was always somebody seen with awe on the podium. And he was one of those researchers who made this terribly confusing new world of genetics very accessible to patients who really did not know what a deletion was, or a mutation, or a heterozygote, or a homozygote. In those days, this was really not even Latin to us, but certainly French, and we weren't conversant like Christina is in French. And Frans was one of those researchers who really made that intricate information so accessible. So I see Frans is back. Thank you very much, Professor Kramers, and for inspiring us as patients from all over the world as to what could be done, and that this information was not the pristine and elevated knowledge only of the professors, but that as patients we needed to know, and you made us aware that this information is accessible to us and that we could understand it. And to both of you, I would like to say congratulations. It honestly couldn't happen to two nicer people, and we thank you for your inspiring and really terrific inspiration to all of us for so many years, and you continue to inspire us today. Thank you very much and congratulations.
Thank you.
Thank you, Claudette, so much for sharing those words. Absolutely. I think making research accessible to all and making it understandable to all is of paramount involvement to making sure everyone is involved and contributing as much as possible. I see two more hands raised to express thank yous. I will ask you to wait until the end where we will have more time to express thank yous. And now that Frans has returned to us, we would like to proceed with the Q&A interview aspect of this evening. So without further ado, I'd like to bring back some old memories of the early days of finding novel retinal disease genes at a time that only the gyrate atrophy gene, the OAT gene, was earlier discovered through biochemical studies. So, Frans, can you tell us a bit about your PhD study in Nijmegen and how you got interested in retinal diseases such as choroideremia?
Yeah, thank you, Fiona. So I hope my computer will not crash again. Anyway, let me just start this quite old story. So I started my PhD in 1986 under the supervision of B. Wieringa and Hans-Hilger Ropers. And I had basically two main goals. First one was to collect all the known DNA fragments from the X chromosome at that time, to get them from all collaborators, and to use them and to map them, to fine map them on the length of the X chromosome. To do this, I used hybrid cell lines that were generated by Hans-Hilger Ropers, and I could position all these 200 markers along the X chromosome. But I also use other DNA. So I also use DNAs from patients that had cytogenetically detectable deletions on the long arm of the X chromosome. And these patients, they would have different conditions. Among them, there were intellectual disability, hearing impairment, and retinal dystrophy, which was, of course, choroideremia on the long arm of the X chromosome. So I could use all these cell lines and DNAs to map 20 DNA markers, these small DNA fragments, to that part of the X chromosome where we already had a suspicion where the choroideremia gene should be located. So that's what I did at that moment.
Thank you, Frans. So we're talking a lot about genes here, and this is something we're quite familiar with now in terms of our scientific language. But at that stage, where was the Human Genome Project? What stage was that project at those times?
Yeah, it was, it was at its infancy, right? So late 80s, 90s, we were all together building the genome. And I think most of you will know that the genome was supposed to be done in 2000. So in the late 80s, 90s, we were building the genome. So that was all a lot of work to get it all together.
Thank you, Frans. So I'd really like to bring Frank in here. So Frank, can you tell us how you got involved in finding the choroideremia gene?
Yes, it started in 1982 when I started as research commissioner of the patient organization of Retina. And by networking, I got in contact also with Professor Pawlowicki from the Institute for Human Genetics in Munster. And at that time, I wanted to leave my hometown in 1984, but he proposed me that we could write together on a grant proposal. And that was so attractive that I skipped my intention to leave the hometown, and especially as he accepted that we include linkage, that we include a choroideremia field study as part of the application. So, and I started to work then in 1985, in January 1985, and we collected data, and the intention of the project was to improve genetic counseling in retinal degenerations, and also, of course, to prepare linkage studies by this field study. And then there was a helpful coffee break in Amsterdam at the Congress in early 1986, and where a discussion with Professor Enright led to the idea that it is possibly a good way to look for microdeletions, and as a kind of shortcut to come quicker to the aim. And the aim was to find the cause of the disease, to isolate the choroideremia gene. But well, as a result of that, first of all, there was a workshop in Bad Nauheim at the Retina International Congress. And we had a workshop with Hans-Hilger Ropers, and I asked at that workshop to the audience, how many individuals from different unrelated families with choroideremia one should collect in order to have a sufficient chance to find a microdeletion? And the answer, as far as I remember, was, well, 30 to 50 families, and they had a number. And from that time, I've chosen a double strategy. First of all, families as big, as large as possible for linkage studies, plus as many as different families with choroideremia, and from each family at least one blood sample from a male person. And so a couple of months later, Frans Cremers called me, and from that time, the two groups in Nijmegen and Munster, they joined forces. And the blood samples, the first blood samples, found their way to Nijmegen. And at that time, I could also meet not only Frans and Hans but also the other staff members in Nijmegen.
Thank you, Frank. So we now have the joining of two superpowers in Frank and Frans. So Frans, back to you. Now that Frank has brought you these blood samples, what did you do with them?
Yeah, he came to our lab, with these 10 blood samples in his bucket. And of course, what we did is we extracted DNA from the blood cells, as we normally would do. And together with Doreen van der Pol, my technician, at those days, my colleagues now will not know the word anymore, but we made southern blots. So we took the DNA, cut them in small pieces, and then put them on a gel to migrate the fragments in different sizes, put them on a filter, hybridize them with a radioactive probe, which nowadays is not done anymore. But that was done to see whether any of the 20 probes that I had already assigned to this region, whether any of these 20 probes would be finding something absent in one of these individuals, thinking that could be something missing. And indeed, it was a huge surprise that in the first 10 samples that we analyzed, 2 of them were missing one of these 20 probes, just one of them. So at that moment, we knew this is a huge step forward.
That's amazing. So now that you've identified these microdeletions, what were the next steps towards actually finding the choroideremia gene itself?
Yeah, so this was a starting point, basically. So we got more blood samples from Frank because he collected dozens of these. We also got blood samples from other places, like from Helsinki, from Eva-Maria Sankila, and we took them all together, and they were used to find more microdeletions in the end. Together with my technician, it took us another 3 years then to take this first probe that was missing in these DNA samples and to see whether we could clone the DNA around this DNA marker. So it took us like 3 years with 2 people full-time to clone a significant piece of DNA of that long arm of the X chromosome. And the next step was, well, can we find in this region, can we find evolutionary conserved elements? Thinking that that's what is conserved may also be important, right? And may encode for a gene. So we took the DNA, and we call this zooblots. We took the DNA from human, chimps, frogs, fish, birds, and we again made southern blots. And one by one, all the DNAs that we had cloned, we would hybridize them on these kind of blots. And if there would be a signal in each of these lanes of all the different species, we would consider, yes, this is conserved. And then we can fish for the gene itself. So we use those fragments, the conserved fragments, to find pieces of the cDNA, the copy DNA. These are the parts that encode for the choroideremia protein. So at that time, around 1990, we had found about 2/3 of the gene in this manner.
That's amazing. And what was so unique, or what was so special about this at the time that you were doing it?
Yeah, so this was all new, right? I mean, so this got published into Nature, right? One of the top journals even at that time. It was published in Nature because it was one of a few. At that moment, only a handful of genes was cloned in this manner, where we did not have any a priori knowledge on the biochemical defect. So at that time, cloning had been achieved already for the Duchenne gene, for the retinoblastoma gene, and for the cystic fibrosis gene. And that's it. So we were, the 4th or the 5th to find such a gene in this manner. And that's why it was so big news, and it had so much attention in the media.
Absolutely, and I think really highlights the pioneering nature of research in IRDs. So Frank, back to you. After all of your work, you must have been very satisfied with these results.
I was especially happy also that we had good news at the Congress of RP International in 1990 in Dublin, and where we spoke to the audience, Frans and me, in two parallel sessions, Frans to the scientific audience and myself to the lay audience, and with many patients attending this session. And I remember me well saying to them, well, listen, at that time I was your vampire, and the only thing I was interested in that was your blood, and you gave it to us, and now we come back with full hands offering you the solution of the problem. And here it is, voila, here is the amino acid sequence of the protein that is missing or rearranged in us, the choroideremia patients. So that was a very good moment. And Frans and me, when we stood later on in front of Trinity College and we discussed about the future, and Frans said, I asked him, and he said, well, there exists a lot of other things, interesting things other than choroideremia. And I think he was very right, and the same was true also for me.
Thank you, Frank. It must have been almost like a dream to give your blood and to get the answer directly back like that. And so to Frans then, how did this discovery change your life?
Yeah, well, it was life-changing, you know, as you may imagine. First of all, I was invited to come to Melbourne, Australia in 1988. It was a Retina international meeting. And this was my first big meeting, my first big flight. So, you know, this was life-changing. I came to Melbourne with a huge jet lag. So I was shaking on my legs, walking through the hallways there. Of course, I met patients for the first time, affected individuals with these conditions. And what I remember very well is that in the evening, we would go out for dinner. And so I would meet persons with Usher syndrome that many of you know, they have hearing impairment and a vision impairment. And these people were leading me through the restaurant. You know, that was like, okay, these people, they have really vision defects, but they were leading me through the restaurant. So that was like, you know, an eye opener for me, what these people could do. Of course, I also met the pioneers in this really small field that was at that time. My presentation, I guess, also brought hope to all people there that, you know, if you can find one of these genes, you may also find all the other genes. And of course, since then, so much has happened. And by now, we know about 200 of these genes, maybe a little bit more. And of course, in the decades following, my team found another 22 of those genes and was involved in finding another 12 of these. So we just went on to find other gene defects. And I think by now, I would estimate that more than 90% of all the underlying genes have been identified since then.
What an incredible impact. And we've already heard from Claudette earlier, who expressed her thank yous to you of the impact that your presentation at these events had in terms of opening this world up to patients. So after finding the choroideremia gene, what was next? What did you do?
Yeah, so, you know, this breakthrough finding, of course, kickstarted my career. So I was happy to receive a award of 5 years, this development award for your career. And it also enabled me just to take my salary and go to the outside world. So I found a nice place in the US with Miguel Seabra in Dallas, Texas. And I took my wife, Diana, and my kids down to Dallas. We spent the whole year there. And I was fortunate because Miguel Seabra at that time independently had already found the function of the choroideremia protein. So I could just work on the biochemistry of the protein for a full year, which was wonderful. Now, Miguel, he worked on his PhD in those days in Dallas. But after a few years being an assistant professor, he moved to London, became a full professor there, and then met Robert McLaren. And together they designed, in the end, a gene therapy for choroideremia, which was also very, awarding for all of us.
Thank you, Frans. And to you, Frank, at this point in the journey after the discovery of the choroideremia gene, what did you do?
But for me, I knew that now the further choroideremia work is in good hands. And for me, it became important to realize that all retinal degenerations are rare diseases. And at that time, also, the EURODIS as European Organization for Rare Diseases, as well as the European Commission, played quite a big role in that decade. It was that the European Commission, for example, who dealt with questions like the orphan drug regulation, but also it was possible to initiate, to co-initiate from here, together with Professor Pawlowicki, a concerted European action prevention of blindness on molecular research and medical care in retinitis pigmentosa, with the aim of developing of promoting the networking of research in Europe. And this took from 1989 to 2001. And at that time, I was also then member of the project management group. It was funded by the European Commission, and we organized 25 congresses on retinal degeneration in 12 European countries, and also exchange of personnel and material. And this was a way to promote, another way of promoting research. And from there on, so it moved afterwards more also back to Germany with the stimulation, or with the stimulus from EURODIS to build national alliances. It was possible also to co-found the German Alliance on Rare Diseases, AXE, Allianz chronischer seltener Erkrankungen, and to work on several projects, also on topics like patient participation in evidence-based patient information, or participation in guideline development and electronic health records, and quite a very different aspects of patient participation, but a growing community of patient organizations in Germany. And another field then opened also in 2004, where I started as patient representative as a federal joint committee, and where I then also became a co-speaker in the field of quality assurance in medical care. So these are various fields of patient participation that were very important, and who showed that the promotion of research and medical care for people with rare retinal degenerations is partly also included in the whole field of rare diseases. And this is of mutual effect and support, and very important.
Thank you, Frank. So question to both of you now. Perhaps I'll come to Frans first, but could you describe the importance of patient involvement in your studies?
Yeah, of course, this was huge. In the last 20 years, I met so many other affected individuals and their parents, and they made crucial contributions to my work. And I just want to give you one anecdotal example, which also brings back memories. In 2007, we had a collaboration set up with Robert Kunekoop from Montreal, Chris Engelhun from Leeds, many other people, Anneke den Hollander and Ronald Roepman from my team. And together we found this rare disease gene, lebercilin. And one of the families in which there were affected kids with this condition, they came to visit us in February of 2007. They came at the day when we had the most severe storm since decades. If I now go out, I can see the trees still lying down in the forest, because that was that severe storm, right? As you now have an island, we had it here. They came through the storm, flying in. They had a personal driver to bring them to Nijmegen. And the next day, so we had dinner together. It was all nice. The next day, they came to the lab and they talked and gave a speech to my team. And that made a huge impression on them and on me. And since then, of course, we have been friends, because, you cannot escape from being friends with these people. And not only they were interested to meet us, they also then went on to do fundraising. And they had a huge impact to get the studies done for this particular gene, and also to get gene therapy done for this gene. Of course, similar stories I can tell you from other parts of my studies. For example, in my Stargardt disease research, that is more recent, we found many hidden mutations in many young patients, which can also lead to new therapies for some of these. And of course, without the financial support in general of all the patient organizations from the UK, from the US, Ireland, Germany, and Netherlands, I could not have done this work, right? So I owe them so much as a whole group.
Thank you, Frans. I think really powerful stories that do truly illustrate the impact and effects that patient relationships in research can have. And Frank, over to you.
Yes, I would like only to add or underline that the importance of the social and intellectual contact between scientists and patient representatives and patient organizations. So in order to think about research aims, in order to develop research plans, and also if there is a project in which fits a work package for a patient organization, it should be also covered in the research plan, in the application. And so I think this interaction is extremely important and can be fruitful when it is well managed. And in addition, the choroideremia example, it shows also the importance of register data, of biobanks, and last but far from least, the importance of congresses like the Retina International Congresses.
Thank you, Frank. And absolutely, these are all values that we stand for here at Retina International and across our international community. So thank you so much for sharing that story. Such a brilliant demonstration, again, of the power between patient and researcher relations. So congratulations to you both on your awards. As mentioned by many esteemed colleagues here this evening, they are truly well-deserved and could not go to more friendly and charismatic gentlemen this evening. So just before we wrap up for this evening, I'd like to extend the opportunity to Frans and Frank. It's such a complex network of colleagues over the years. I'm sure you have many thank yous to offer. So Frans, I'd like to invite you to extend any words of congratulations or thank yous you'd like to share.
Thank you, Fiona. Yeah, of course, I share my thank you to Frank first of all, but yeah, let me just start with Retina International itself. I've been inspired by the unrestricted support of all people from Retina International, foremost Christina Vasser, in all these years for my research. She always supported my sometimes too ambitious plans. Likewise, Frans Bedura, then working with Retina, always supported me in my small efforts to get something going. And he was often the person that could start and these pilot studies that then would lead to a much larger study in the end. Of course, I want to thank many other people, but I don't know where to start. For my initial work on the choroideremia gene, apart from Frank, which of course I owe everything tonight, my technician Doreen van der Pol and my mentors B. Wieringa and Hans-Hilger Ropers. For my subsequent work, of course, I was blessed by many talented colleagues that supported my studies. And I just want to mention a few: Ronald Roepman, Anneke den Hollander, and Camille Boon. They have been doing their stuff independently already for a long time. I could also build, on their shoulders work. My recent Stargardt disease work would not have been possible without my very talented colleague, Susanne Roosink, who's supervising all the non-Stargardt studies. Also the clinical work of Ingeborg van den Born and Karel Hoying, the help of Alex Garanto and Rob Collin, and a huge amount of work performed by my talented former PhD candidates, Ricardo San Germano, Mobin Khan, and Esme Runnert. They were also crucial for me. And then coming back to the choroideremia gene, this was a cloning race in those days. It was also inspiring by competing with UK and US groups. And of course, it was inspiring because we won the race in the end. But funny enough, what happened is over the years, the competition changed over the years to collaborations with many colleagues worldwide. Too many to mention here, but I just want to say and mention a few people that are, that have a special place in my heart now and forever, which are Robert Koenekop in Montreal, Mirjam Bouwens, Frauke Koppieters, Elfriede de Beer in Ghent, Claire-Marie de Hanens in Lille, and Rande Alkmets in New York. With such friends, you will always do fine. And finally, my last word of course goes to thank my wife, Diana. I love you. Without you, this journey would not have been possible, for sure. Thank you very much.
Thank you, Frans. And Frank, over to you.
Yes. So I start also with Christina and Frans and Retina International with my thanks, and who has listened today also the words of Christina and Frans. You may, even if you don't know very much from Retina International, you may have experienced here at these moments also something of what Retina International and its activities mean. So thank you, Christina, for all that, and Frans. I would like to thank Reinhard von Geschitzky and Maria Kretschmer for in the early times of research promotion, and then of course all patients with choroideremia who gave their information and were willing to share blood samples, to give blood samples, and apart from Frank, these are the main contributors to the success, then, in a way. And then I mentioned several times also Iva Pavlovitsky, who played a key role in my career and opened the institute for me, and we went many ways in Europe together. My thanks are to Herman Krastel, not only for supporting the choroideremia field study, but also for developing the video campimeter together with my brother Jörg Brunsmann and myself, and for Ulrich Kellner and Klaus Gehrig for coping with everyday questions we are faced with at Retina in the field of medical care. I'm thankful for Klaus Rüther and Norbert Ahlers for ethical discussions and questions, which led finally also to a publication in The Lancet. And I'm thankful for the staff of Eurodis, and especially also the staff of AXE, of the German Alliance for Rare Diseases, with Christoph Nachtigeller, Miriam Mann, and Christine Mundlos, and Hartmut Fels, and others in developing the lobbying in the field of rare diseases in Germany. And I'm thankful for Alfred Hildebrand, who supported many projects on this way on the field of rare diseases in Germany. And then I'm thankful for, just let me wait a moment. Yes. So I'm thankful for in the field of the work of the Federal Joint Committee. I'm thankful for Cordula Mühr, Sabine Häfner, and Jana Hennings. And in the they are my close colleagues, and I'm very happy that here in the field of patient representation together with you. And finally, I'm thankful for Alicia Rubalco. This is my wife, and she helped me in the last 20 years, day for day, in all moments, and she always insisted not to be mentioned in any publication. And thank you very much. And I say to her, like Frans said before, he has stolen from me, but it is the same. Alice, I love you.
Thank you, Frank. It is truly through our networks that make us stronger when we work together. I'd like to now just reopen the floor to our attendees here today, anyone who would like to express words of recognition. I'm going to invite Christoph Nachtgaller, who is the president of AXA. I know he is looking to express his words of recognition to Frank this evening. Christoph, over to you.
Yes, I hope you can hear me.
We can.
Wonderful. And do you see me too?
We do not.
Oh, you do not? What can I do to?
Alt V.
That's what I'm always afraid of. I see everybody of you, but well, the best is that I begin to talk. I think that's the most important thing now. Dear Frank Brunsmann, dear colleagues, dear Avril, I see you since a long time. Once more, we met in Brussels and in Venice and everywhere in the world. So I wanted to say some words of recognition and thanks to Frank Brunsmann. I'm the former chairman, and now I'm the honorary chairman of AXA. And so it's my pleasure to extend our warmest congratulations on behalf of AXA to Dr. Brunsmann on this wonderful award. He is a co-founder, together with me and others, of AXA, the Alliance of People Affected with Rare Diseases in Germany. And we established the organization in 2004. And since then, he has supported and represented the overarching interests of those affected by rare diseases in many ways. One of his key issues is the development of criteria for patient-oriented disease descriptions, especially with regard to evidence and quality assurance. And he has supported many of our member organizations in preparing such descriptions for their diseases. Thanks from them too, dear Frank. His recommendations were also included in our big German National Action Plan for people with rare diseases and are still receiving attention. Dr. Brunsmann is also a great advocate of patient participation and has already developed concrete proposals for framing patient collaboration. And he is and was an advisor to the AXA Executive Board, to me and to others, and we work together in many projects. And he continues to provide us with advice and support today. Today, we are very pleased about this. And I have a wonderful, we have a wonderful cooperation, and I would say friendship together, and this developed over many, many years. And in this sense, my best congratulations, and on this way, hugs and love, dear Frank, from us all, and from me personally, Christoph from Düsseldorf, from Germany. congratulations.
Thank you,
Thank you very
Christoph, very
much.
much. Thank you so much.
Thank you, Christoph. I would like to invite Reinald von Gizicki, please, if you would like to unmute. Reinald, if you would like to click Alt-A to unmute. There we are.
All right, can you hear me?
You can.
Dear Frank, dear all, I am very, very happy to be part of this ceremony, which also for me is a kind of remembrance meeting, to be in together with Claudette, I think she's here as well, and Christina and Christoph Nachtigaller. But Frank, when listening to all this, in particular relationship with Frans Cremers, I wondered if I were present as well, because I was in a way, but on another level, I think. You were the more science-oriented person. You were there when Retina was founded in or was going on towards the conference in '86 in Bad Nauheim, and you were a research commissioner, always very close to research, and even so close that you were looking into the genes for your own disease. You were, if I remember right, you were diagnosed in the beginning for gyrate atrophy. And gyrate atrophy, I think at that time, was thought to be treatable by ornithine or arginine. So, and then you were happy to find out you had this disease, but then you were unhappy to find out you had another one, which was much, much more serious, choroideremia. And that, I think, stimulated you very, very much. And your science orientation, going together with patient orientation, made you a great mediator between both communities. You were publishing for both sides. You were publishing, I remember in Nauheim, Patients Fight for Sight. At this, I wasn't aware, although I was at that conference, that there were so many things you were talking to the scientists as well as to the patients. That is the two faces which make patient-oriented research so worthwhile. You had that all the time. And I think you stimulated not only Frans Cremers, but all the people around you, like me as well. As a, I remember in many conferences where we were together, you were in my room somehow, and we were talking somehow, and I didn't even know that you were there, but there you stimulated me and others around. And I very much remember the last activities of yours with AXE, and then with the various groups inside Retina, which helped us to found and develop the social environment so stimulating for patients as well as scientists. So I'm very, very grateful. I thank you very much, and a very big hug to you, Frank.
Thank you, Reinald.
Thank you, Reinald. I'd like to invite Robert Quinnecoop, if you would like to unmute, Robert.
Yes, it's working.
It is working.
Thank you very much for inviting me to come to this wonderful celebration. I want to thank Retina International for all they've done for all these years, and also for putting together this wonderful event, and for choosing Frank and Frans. And then I would like to specifically congratulate Frank and Frans for their achievements. I've always been very inspired by Frans Cremers ever since I met him at Arvo in 1995. And I already knew about his choroideremia groundbreaking work, and I knew about Kramers. And one of the wonderful things about research many things are wonderful about research, but one of them is that you can pick your own collaborator. And I definitely wanted to pick Frans to work with. And so we started working together in 1995, 1996. And with Anneke den Hollander, we made some amazing discoveries. We discovered the CEP290 gene. And now today I'm injecting the antisense oligonucleotide for the CEP290 mutation that Frans, Anneke, and I discovered. We're treating these patients. So this is a very wonderful day for celebrating their achievements, exemplified by work that we're doing today in Montreal, where, by the way, it's -20. So what happened with Frans and I is that we chose each other to work with, which is a wonderful thing. And it just grew and grew. And now we have developed a friendship, a lifelong friendship, as Frans said. And now we share interests like weather patterns, archaeology, biking, especially Dutch bikers, and of course, volcano eruptions and archaeology and other things. So we're still feeling each other out what our common interests are. And we are also planning our retirement together. So it's been a wonderful journey, Frans. Thank you so much. And congratulations also to Frank.
Thank you, Rob.
Thank you, Rob. Such a wonderful story. So we are just coming to time. I will invite two more people just to express very quickly their thanks. So first will be Neil Bench, and then I'll come to Juliana Salam after. So Neil, over to you.
Hello, can you hear me?
Yes, we can.
Hi, I'm Neil Bench. I'm the current president of the choroideremia Research Foundation, and I'd just like to very quickly thank Frans and Frank for all they've done throughout their careers. And I'm speaking for all the members of the choroideremia Research Foundation and their families. And we wish you the best in the future. And thanks again. That's all.
Thank you, Neil. And finally to Juliana. If you'd like to unmute, Juliana.
Hello, can you hear me?
Yes.
Yes. So Frans, when I saw you were the one that is going to receive the award, I say I should stop by and say thank you very much for everything you have done to the research in the field of Stargardt patients. I have been I had the pleasure to be with you to discuss in many meetings. You were so open and you built up opportunity for patients from Brazil. So thank you very much. Exactly what I was going to say is the same that Robert Kunakup said, but he always says better than I with my poor Latin American English. So the only difference is that we have 30 degrees around here. All the rest, the feeling is the same. So thank you very much. And thank you also, Frank, for developing the choroideremia research. I am PI for the choroideremia trial and for the CEP290 trial in Brazil. So that is a fruit of the work of you too. So thank you very much.
Thank you, Juliana.
Thank you, Juliana. You're all very obvious.
May I add something?
Of course, Frank.
Yes, I would like to add that well, there are, first of all, thank you for all those who thanked, and I can customize this situation. It is very fine. But what I want to say is, well, that the ways of research promotion at that time, they are different from the ways that are fitting today. Each time has its challenges, yes. And but what I would like is that we find ways to really learn from each other, like Frans and me this time when we prepared this meeting, and I learned very much from Frans in the way he wrote. He prepared this little ceremony, and I prepared myself. So we learned from each other, and so to find a good way, and this I would like that Retina International takes the chance in the next and the over next and at the meetings at Retina International to cope with the questions of today, like, for example, to find a good idea about what are the right endpoints to include in clinical trials, yes, and to have a common patient perspective on this question. These are things that are not yet answered, and every time has its questions, and to ask the right questions. This would be my general remark from me. Yes.
Thank you, Frank. Truly, the work is not yet done, and the onus is on us as a global community to keep answering these questions to improve the lives of those living with retinal degenerations and to find a cure for each of them. So that will bring us to the end of today's awards ceremony. I would like to extend a really sincere thank you to our contributors today. We have our CEO, Avril Daly, we have our current chair, Frans Badura, and we have our former president, Ms. Christina Faster, contributing today. Thank you for your time and for your words. And of course, our final congratulations and words of recognition must go to Frans and Frank. Thank you for sharing your story and your energy today. And of course, thank you to our attendees for all your contributions and for coming and for listening. We look forward to welcoming you to our next Retina International event, which will be advertised shortly for World Health Day. And we hope you enjoy all the rest of the events happening this Rare Disease Week. Thank you very much.
Fiona, before closing, finally, let me say one sentence, please. This is normally the point at a meeting after such a really great ceremonial words have been done, and two persons who really deserved that honor. I will take my trumpet and play a little bit for you. I promise both of you, I will do it when we meet in person. Instead of that, I was catching 2 glasses of champagne and I make a
Good, fine.
Congratulations to both of you, and thank you very much on behalf of the whole Retina International family, and goodbye to everybody.
Thank you, Frans. Thank you, Fiona. Thank you all.
Thank you very much.
Thank you, Fiona.
And the trumpet is promised, right, Frank?
It's promised.
We'll hold you to it. Thank you, everybody, and take care.
Bye-bye.
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 Thank you all.
Thank you very much. Bye-bye.